you've now spent a couple of days looking at and watching some examples of how proteins can actually be made from our DNA. We've already referenced that and called it the central dogma of biology. Gave two reasons for that. One is that every living thing will follow this process, but it's also very central to life's processes. Our cells have to be able to make proteins, and the instructions to make those proteins actually come from our DNA. Now, our DNA is much too valuable of a molecule to ever leave its nucleus in our eukaryotic cell. But it is the ribosome floating here in the cytoplasm where the proteins will actually be made. So we have to find a way to send a message from the DNA out to the ribosome so that the ribosome can make the protein. So our central dogma is the idea that we're going to start with a piece of DNA, and then somehow, some way, those instructions need to be followed in order to finally produce what would be our amino acid chain or our protein. Now, how we actually get to that requires two steps. So, first of all, we're going to start with our DNA, and our DNA is going to be transcribed. That is the name of the process. So the first thing that is going to happen is called transcription. And in transcription, we are going to use the DNA and build a messenger RNA molecule. So this messenger RNA is what will be allowed to leave the nucleus and head for a ribosome. This messenger RNA, mRNA, is just that. It's a message. So transcription, we build a new set of instructions called messenger RNA, and that's what we're going to look at today, is exactly how we do that. From the messenger RNA, it's going to leave the nucleus and head for a ribosome. Now let's look at a close-up view of a ribosome here. In the ribosome, we have a large subunit and a small subunit, and the messenger RNA is actually going to travel directly between those two. So your messenger RNA, when it reaches the ribosome, is going to go through our second process, and that process is called translation. So first we had transcription, and now we have translation. So your ribosome is going to read this messenger RNA in a very particular way. And as the messenger RNA is read, your ribosome is going to begin bonding amino acids together. So if we take this process from beginning to end, we started with an instruction in the DNA. We built a transcript or a rewrite in our messenger RNA. That was able to leave the nucleus and head for the ribosomes, where the ribosomes could translate the RNA very carefully to build an amino acid chain. When we talk about the cell's ability to make a protein, typically a protein comes from the following of the instructions from a gene. So we do know that genes are small segments of a chromosome. So a gene is a segment of DNA. So you can see here, I've color-coded so that we can see some different genes. And you can see that these genes are made up of A's and T's, G's and C's. So our job is going to be to use today this DNA. We're going to transcribe that DNA into messenger RNA and also follow steps in order to be able to decode that messenger RNA like you are the ribosome and build a protein. I've simplified our situation, and we're just looking at now one gene from our nucleus. So in this DNA, let's not forget that between the two sides of our DNA, our bases would be connected by hydrogen bonds. So in reality, between every complementary base pair, there are hydrogen bonds, and we already know that from our previous studies of nucleic acids. Now, we also know that when we do have our double-stranded DNA, there are enzymes that are able to break those bonds, and that enzyme is helicase. So in my little example, I have a pair of scissors that are going to function as helicase. So I'm going to use the helicase. I'm going to break these hydrogen bonds so that I can essentially access this template of base pairs inside. This might look and seem very similar to replication, and there are some similarities, but we're also going to see some differences. The helicase has actually now done its job, and it has broken the bonds, the hydrogen bonds, between our complementary bases, which allows our DNA to essentially open up. So we now have access to the bases as a template, essentially, and from that template, we will be able to build our messenger RNA. Now, messenger RNA has a similar base pattern as to what we saw when we used and built more DNA. Our T's are going to bond to A's. So if there's a thymine on my template strand, as we build the messenger RNA, our base that would be brought in would be an adenine. Adenine, though, on our template strand, will actually bond with what is called uracil. So we do have a slight difference in our base pairings. Cytosine still bonds to guanine, so I can follow this piece of DNA and I can match every base pair. I have a guanine on my template strand. I'm going to get cytosine on my RNA strand. That base pair is still the same. Adenine, goes to uracil. And as you can see, I'm simply following our base pair rules to fill in my messenger RNA. Made a mistake. No thymine. We're making RNA. We need a uracil. So cytosine, guanine, uracil, adenine, etc. So what's going to happen in some future examples, you are going to determine your messenger RNA strand. You'll be able to choose the appropriate answer here in the Edpuzzle, and then you'll be able to see whether or not you did that correctly. So we are right now talking about transcription as we take DNA and write a slightly different version of those instructions as messenger RNA. And we do again have our base pairing rules. So with transcription, if there is a cytosine or a guanine, those will pair up. An adenine on the DNA will give us a uracil in our RNA, but a thymine will still give us an adenine. So we're going to do some more examples of this, but you can always go back and rewatch this segment to look at how it is that I came up with what would be our messenger RNA strand. So remember, this is what would have been in the nucleus, and that messenger RNA strand will be going to the ribosome for the next step of translation. We're now ready to look at our messenger RNA as it moves its way from the nucleus to the ribosome. So this particular messenger RNA is going to make its way and travel between the two subunits. Now it turns out that it's almost like decoding a secret message. The ribosome knows how to do it. We just have to learn what the ribosome already knows. Now it turns out that the ribosome decodes the messenger RNA 3 bases at a time. But there is a very special place where the ribosome always wants to start. The ribosome is always going to start at an AUG, that's our 3 bases, your ribosome is always going to start at an AUG. So, as I look at our messenger RNA, it turns out that I had us set up for success here, and I have an AUG right at the top of our messenger RNA. So I'm going to go ahead, because I know the ribosome is going to work in 3s, and I'm going to group 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. I know that this is how the ribosome is going to go about decoding this message. Now, biologists have created a couple of tools for us that can make decoding this message a little easier. So I'm going to bring a document in here. You have a copy of this on your Google Classroom. And both of these documents will give you the same results. Some of you are going to end up liking the circular version. Some of you are going to end up liking the square version. It doesn't matter to me which version you use. But all we have to do is follow our 3 letters at a time. So AUG. We'll use the circle one first. You start in the middle. So our first base is A. Our second base is U. So if you notice, I worked my way out from the circle, A to U. And our third base is G. So I worked my way out a little farther. So if my codon, that's the 3 letters, is an A, U, G, then the amino acid that your ribosome is going to be looking for is called methionine. Now, we don't need to write out that entire name. We get to use an abbreviation. So it turns out that our first amino acid is going to be methionine. So we've always used a circle for an amino acid. So I'm going to put a circle with MET in between. That's my first amino acid. So go back to my chart, figure out the next amino acid. Well, this says it's G, G, G. Those are the 3 bases that I've grouped together. So I start with my G in the center, G, 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 first base, second base, the third base, and I get glycine. So my ribosome would bond to the methionine, the next amino acid, which will be glycine. And this is how the ribosome does it. We get to use this little tool to help us decide which amino acid comes next. Let's try the square block. We have a CUA. All right, so if you use the square version, your first base, notice it says first position. So our first position was a C, so that means I need this block. Our second base is a U, so I'm going to look at the top for the U's. So the C and the U, where those intersect right here is where I'm going to find my CUA. And if I zoom myself in, CUA is an amino acid called leucine. Now, this one already has been abbreviated for you. Leucine is LEU. So my ribosome would bond a leucine amino acid next. Please don't panic. You will never have to memorize those charts. These are tools that you will always get to use. All right, I'll take one more slowly for you. AAG. I'll use the square version. A is my first base. A was also my second base. So I'm looking for where my A's meet, which is right here. AAG tells me it's going to be lysine. Now, just in case you're curious, if I come up here, AAG tells me lysine. It does not matter which of those 2 tools you use. It's going to be up to your preference. My cat wants in on the deal. All right, if I move along and try to finish out my protein, My next codon is CGG. I'll use this one this time. CGG is arginine. So I will add an arginine to my protein chain. UCG, UCG says serine. So I'm adding another amino acid, and I have a UAG, just to move it around, UAG, and oh my goodness, UAG is an amino acid that says stop. So this would be telling the ribosome this is where the protein would stop. So it turns out that this particular section of DNA, this gene that we had from the nucleus, when we transcribed it and then we translated it, we ended up with a 1, 2, 3, 4, 5, 6 amino acid protein. And for now, we don't know what this protein is really going to do, what it might mean, what trait it might be, but we were able to start with our piece of DNA, transcribe it to our messenger RNA, and then translate it to our protein. These next examples will pause throughout the Edpuzzle. So I will start you off with your DNA. I will open it up or break it open with the helicase. You will determine your own messenger RNA. You'll choose an answer in the Edpuzzle. And then you'll be able to also translate your messenger RNA, and you'll choose an answer as well for your protein. So here is your next piece of DNA. So as we unzip it, the helicase has now opened the DNA. So I would encourage you to have a pencil ready. When the video stops, please determine your messenger RNA. Here was your correct messenger RNA. Now I'd like to remind you that before we move on to translating this messenger RNA, we have to make sure that we always look for our start codon. Your ribosome will only start at an AUG. So if I start at the top and I look for the sequence of AUG, I find that AUG a few bases in. So as I start to translate, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So using your correct messenger RNA, please translate the protein. I'll walk you through this again. Starting with an AUG, if you choose to use the tool at the top and you start with an A in the middle, to a U, to a G, the first amino acid is methionine. So your ribosome is going to begin with an MET, or a methionine amino acid for your protein. Your next codon is GUA. If I use the square this time, just to mix it up, my first base is a G, my second base was a U, my third base is an A. So GU, that means it intersects down here. So I will find GUA. It says that amino acid is valine. So my second amino acid, methionine, is bonded to valine. For AAA, A to A to A gives me lysine. There's my third one, my third amino acid. And UGA, UGA is again a stop codon. So this particular strand of DNA resulted in a protein that only had 3 amino acids. And again, we used either the circular or the square tool to actually be able to decode the messenger RNA, just like your ribosome does. Here's your second independent example. We have a piece of DNA. Helicase is going to again break the hydrogen bonds so that we can access the complementary base pairs. from one side of the DNA, please transcribe and build your messenger RNA strand. Now that you have a properly transcribed messenger RNA, please don't forget your start codon is AUG. So scanning from the top, I have an AUG. Please also don't forget your ribosome is going to translate or decode this message 3 codons at a time. So I strongly recommend that you block off each codon in order to use your tool and determine your amino acid chain. This particular trait, as we can see, ended up with 1, 2, 3, 4, 5 amino acids. We ended with a stop codon. So in translation, we have methionine bonded to arginine, bonded to serine, bonded to tryptophan, and bonded to arginine. Your ribosome, don't forget that these bonds it's making are called peptide bonds. It is a special bond that is produced only by the ribosome as it builds your proteins. This makes perfect. Let's do another one. Our helicase, again, has broken the hydrogen bonds, so our DNA can unzip. From that unzipped DNA, we can now use those template bases, and from those bases, transcribe a messenger RNA. As you're transcribing, please don't forget that there are no thymines in messenger RNA, only uracils. So you will not have any T's in your messenger RNA strand, only U's. So an adenine now bonds to a uracil. That's one of our new base pairs. I'd also like to point out that after your messenger RNA is finished, and it's going to move off to the ribosome, so I'll take it out of here as it moves to the cytoplasm, your DNA will actually be allowed to bond itself back together. So your DNA only unzips when it's necessary. It will unzip to make messenger RNA, your transcription. We've also seen it unzip like this before during DNA replication. We have our needed messenger RNA. It is going to move over to the ribosome in order to be translated. Always again, don't forget, your start codon is AUG. Here's your final protein from our green strip of DNA. We have 4 amino acids, methionine, proline, valine, and glutamine. And finally, we had our stop codon. I have one last example for you. This ties into yesterday. When you looked at the firefly in bioluminescence, it mentioned that the enzyme you were making, enzymes or proteins, the enzyme was luciferase or luciferase. And it actually has a total of 547 amino acids in its protein. So these amino acids, these amino acid chains can get really long. 547 really isn't even a large protein. But I did a little bit of research and I actually found the correct and accurate DNA code that would actually translate for the first 10 amino acids in our luciferase enzyme. So I'm going to zoom back a little bit. That's only 10 amino acids, which means it takes 30 base pair. So if I were to transcribe and translate A, U, G, G, A, G, A, U, can't miss that. Ooh, A, U, A. Phew! Let's hope that I didn't make any mistakes. Our next step would, of course, be to look for our codons. We have to find our start codon. That would be AUG. There it is. I think it's always wise to go ahead and mark your codons for yourself. And off we go. AUG will be a methionine. If I'm continuing to look at my tool, GAA is glutamic acid. I hope you are trying to do this at the same time. GAU is aspartic acid. GCA, alanine. AAA is lysine. AAU, asparagine. AUU, isoleucine. AUG is another methionine. Methionine is just another amino acid, so you can find methionine within the middle of a protein, not just at the start. Looks like we're almost done with our first 10. CAC is going to be histidine, and GGU is glycine. Now notice we did not find a stop codon because this protein continues. Again, this was just 10 amino acids out of a total of 547 that will make that enzyme luciferase, which is the enzyme that allows the firefly's tail to have that bioluminescence. Now, right about now is usually when people start to ask, well, what if something about the DNA changed? What if there was a mutation? What if this was a different amino acid because something happened to your DNA? There are a variety of different types of mutations. Some mutations will cause a change to the protein, which could be good, it could be bad. But some DNA mutations we will even find will have no effect. So next week, we will be looking still at how messenger RNA is going to be the result of transcription. So we will be doing more of this next week. So from DNA to our messenger RNA was called transcription. Our messenger RNA being read by the ribosome was called translation. And the ribosome is the organelle that will be bonding your amino acids together, creating peptide bonds in between your amino acids, which, by the way, is also why proteins are also sometimes called polypeptides. Poly means many. Peptides is referring to our peptide bonds. So a protein can often be called a polypeptide. So I hope this went well. I hope that you are understanding our base pairing rules as we match DNA to build messenger RNA. And then also you're getting more comfortable with how to use your tool to decode messages just like the ribosome does. Next week, we will work on mutating our DNA to see what impact that might have on our proteins.